In this video, we will showcase the powerful features and functions of the CeleViz browser. After downloading and installing the software, launch CeleViz browser to access the user interface. The first step is importing data files. CeleViz browser currently supports files in H5AT format. H5AT files can be generated after Singleron's automatic annotation or manual annotation process. It stores information such as quality control, cell clustering, and differential gene expression. Click the File menu in the top left corner and select Open File. Follow the prompts to upload the data. You will then be redirected to the first section, View Data. Here, you can gain an initial grasp of the dataset, setting the groundwork for further exploration. Begin by examining the dimensional reduction plot, which shows the different cell types in the data. The software supports various visualization options, including tsne, map, and their 3D formats. Here, we are showing a 3D map plot. CeleViz browser's interactive capabilities enable you to delve deeper into each plot. You can view sample information, adjust views, fine-tune parameters, and save images. Hover your mouse over an area of interest, you can instantly view the values and information associated with each data point. In 2D view, you can zoom in by left-clicking and selecting an area, and reset views by double-clicking. In 3D view, you can scroll to zoom in and out. You can rotate the view by dragging with the left mouse button or move the view by dragging with the right mouse button. Now, let's return to the map 2D view. Besides its powerful interactive features, CeleViz browser excels in data processing, enabling the users to explore and to present the biological features of the data effectively. The software facilitates selective data display and exploration, as well as highlighting or hiding specific information within a region of interest. This functionality is accessed through the checkboxes on the left side. By clicking the checkbox next to a cell type name, you can highlight the corresponding cell type, which will be visually emphasized in black in the map plot. Conversely, if you want to hide a particular cell type, you can uncheck the corresponding checkbox on the right. Moreover, CeleViz browser provides the option to display data not only by cell type but also by samples. Under Sample Group, you can choose to display the data according to user-defined categories such as disease subtypes, patient ID, or other information. For example, if you wish to visualize the UMAP plot colored by disease group, simply select the Color By option in the top right corner, choose Disease Subtype from Sample Groups menu, and the plot will be updated accordingly. You can also use the checkboxes on the left to highlight or hide data from specific groups. For example, you can easily highlight the disease subtype of interest. One key challenge in single-cell data visualization is customization. CeleViz addresses this by providing personalized parameter adjustment functions, which enables researchers to easily enhance the visual appeal of graphs. These features can be found in the Customize menu in the top right corner. Here, you can adjust dot size, opacity, axis length, font size, label placement, and color palettes. After selecting a color palette, you have the ability to further adjust individual label colors for each cell type. Click Apply to confirm the changes. Finally, you can click the camera icon in the top right corner to save the image. CeleViz browser supports multiple image formats, providing flexibility for your needs. Below the dimensional reduction plot, you can find a list of differentially expressed genes for each cell type within the dataset. The software provides flexible ranking options for various parameters. You can reorder the list based on p-value, adjusted p-value, log fold change, and percentage. Additionally, CeleViz browser supports searching for genes of interest. For example, by entering the gene name CD37, you can view its expression pattern. By selecting B cells and sorting by log FC in descending order, you can identify the top 3 differentially expressed genes, CD79A, MS4A1, and CD79B. For a more in-depth exploration of these expression patterns, let's move on to the next section. Section 2, Gene Expression. Here, we can see a search bar at the top where we can enter the names of genes of interest. We can view their normalized counts across different cell types or sample groups, as well as their expression patterns in raw counts. Let's enter the names of the 3 genes we previously identified and click the Run button on the right. Feature plots and violin plots for each gene are generated. In addition, the software also displays differential expression patterns of genes of interest across different groups. You can apply filters to display data from specific disease subtypes or sample types. Here, we have chosen both the disease and normal groups for analysis. Moreover, in the Groups box, we can choose to organize the data by patient, sample, or other conditions. For this demo, we will group the data by sample, and will compare the gene expression between the disease and normal groups. Click Apply to initiate the analysis. After completing the analysis, a heat map and dot plot are generated. We can clearly see that the expression levels of these 3 genes are notably higher in sample C4 and C5. Furthermore, we can compare the gene expression patterns between 2 user-defined groups. In this case, we will compare the disease and normal groups. Click Run to generate the differential expression heat map, dot plot, and violin plot. Section 3, Cellular Composition. This section allows us to explore the composition of cell types or clusters across different samples within the dataset. The composition can be visualized through pie charts and bar charts. You can select individual samples to view the corresponding proportions. The pie chart shows the cell counts and proportions of each cell type. On the other hand, the bar chart supports data filtering. For example, here we choose to focus on the disease group, and to display the data by samples. The bar chart displays the proportions of cell types, with the sample names on the x-axis. Since we selected only the disease group, the x-axis includes samples C1 to C5 from the disease group. Additionally, the bar chart allows you to hide specific cell types or groups. For example, if you don't want to show information about B cells, you can simply uncheck the checkbox for that cell type on the left and click Recalculate to update the chart. Furthermore, we can compare cell compositions of 2 user-defined groups. Here, we once again select the disease and normal groups. Click Run. We can see the proportions of each cell type in these 2 groups, and if the differences are statistically significant. Section 4, Differential Gene Expression. This section is mainly designed for exploring differentially expressed genes. It displays the top 3 differentially expressed gene heat maps within each cell type. You can toggle between cell types and clusters using the drop-down menu at the top. You can also compare user-defined groups. For example, we can once again select the disease and normal groups and click Run to perform intergroup comparisons. Please note that calculating differentially expressed genes may take some time depending on the size of the data and your computer's processing capacity. Once the analysis is completed, you'll be able to observe the differentially expressed genes across the groups. You can download all the information by clicking the download icon on the top right corner. You may scroll down to view the respective volcano plots. Section 5, Quality Control. Here, we can compare the major QC matrix including mitochondria proportions and UMI counts across different groups, such as cell types and sample groups. You can choose the groups to be compared using the drop-down menu at the top. For example, you can choose to display the quality control information by sample. Section 6, Enrichment Analysis. Here, you can select differentially expressed genes in the dataset for gene ontology enrichment analysis. The results are presented mainly in bar charts and dot plots. Additionally, the software supports uploading custom DA lists in CSV or text format. Upload a gene set by clicking Upload Gene Sets. The entire list of enrichment analysis results can be downloaded by clicking the Download button located at the top right corner. If you find that there are too many or too few pathways in the list, you can adjust the analysis based on log-FC, adjusted p-value, and PCT values, and rerun the analysis. Section 7, Similarity Analysis. In this section, you can select multiple cell types or clusters within the dataset to explore the intersections in their gene expression. The results are presented in Venn diagrams and upset plots. As an example, let's select 5 cell types of interest. The left side of the upset plot shows the number of genes for each cell type. The lower right part shows the intersection in gene expression between different cell types. Solid dots indicate related cell types, and when 2 solid circles are connected, it indicates the shared gene expression. The numbers above the bars indicates the number of genes shared by these cell types. It's important to note that when more than 5 cell types are selected, Venn diagram will not be displayed due to its inherent limitations. The upset plots are in general better suited for the analysis of more cell types. Additionally, you can upload your own list of gene for the analysis. Calculations will be performed after uploading and the results will be displayed. In summary, CeleViz browser is a powerful and user-friendly software for visualizing and analyzing single-cell sequencing data. Whether you are a beginner or a seasoned single-cell expert, you can use CeleViz browser to explore and analyze single-cell data, understand cellular heterogeneity, and uncover biological insights. If you are looking for a solution for single-cell data analysis and visualization, CeleViz browser is an essential tool. Moreover, stay tuned as more features are being released for CeleViz browser.